Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. Before we jump into this new episode, we'd like to take a moment to share some exciting news with you. We are working on a new and exciting project. We want to bring the Dr. GPCR ecosystem even closer in one place and make it available at the tip of your fingers. Soon, you'll be able to access GPCR focused information faster, interact better, and advance your work more collaboratively. If you're as excited as we are, you can sign up to be an ecosystem beta tester. All you have to do is go to drgpcr.com/ecosystem and enter your information. Stay tuned, more details will be released soon. Make sure that you mark your calendar for the third edition of the Dr. GPCR Summit. This year, the summit will be held between October 10th and 16th. Stay tuned, as we are also working on the program for the summit. Visit drgpcr.com to find out more details about all our activities. And now, let's dive into this episode. Hello, everyone. This is Yamina from Dr. GPCR. And here we are yet for another recording, another episode of the Dr. GPCR podcast. Tonight, I have the pleasure of having with me Dr. Khaled Abdelrahman from the University of Ottawa. Hi, Khaled.
Hi, how are you?
I'm great. It's so great that we finally got to do this.
Yes, finally.
Super excited. We were just talking before starting the recording that it's been a while since we bumped into each other at a Great Lakes GPCR meeting, but thanks to COVID, but still, it's going to happen soon, hopefully.
Yeah, you have no idea how much I miss those meetings and all the conversations and interactions with people and the science in this meeting. But we were hoping to have it back next year. I'm not sure with Omicron that this will be possible. We'll see.
I'm really hoping that 15 mutations will make the virus, although more, that spreads more easily, but less deadly.
Yeah, I hope so as well.
Super, super. So let's start at the beginning. If you can give us, please, an introduction about who you are, what is it that you do, and how did you get where you are today?
That's great. That's a long story. So I graduated from the School of Pharmacy at Alexandria University in Egypt. That was in 2006. And because I was I scored second on my class, so I was invited by the faculty of pharmacy to for an instructor position at the Department of Pharmacology. So I assumed that role in 2006, and during this, while I was doing this, I was as well provided, a fully funded master's studentship to do my master in the same department. So my duties were, a mix between teaching and doing research when there's time. And in 2009, I was able to finish my master. My master was focused on how to understand the mechanisms behind the protective effect of some drugs used for diabetes, anti-diabetic medication, against nephrotoxicity caused by some immunosuppressants. As you can tell, like it's faculty of pharmacy, so the master was heavily focused on drugs and side effects and things like this. During that master, I did a lot of vascular biology techniques, so measuring the activity of blood vessels, measuring blood pressure, the kidney function. All these things were very interesting at the time, but I was very interested in the brain, and we did not have the facilities to study the blood vessels, the tiny blood vessels in the brain. So I decided to move to Canada for a Ph.D. So I moved to University of Calgary 2010 for a Ph.D. I joined the lab of Dr. Bill Cole, University of Calgary. And Bill was doing a lot of studying the reactivity of the blood vessels supplying the brain and what are the mechanisms responsible for the contraction of these blood vessels in response to an increase in blood pressure or increase in intravascular blood flow. So it's it is it was looking into specific mechanisms that contribute to the if there is a change in the blood flow in a certain or the blood pressure, how the small arteries, the very small arteries, what we call the resistance arteries, supplying the brain will respond to that change in blood flow. And that's the very important mechanism to protect the essential organs from rush and blood flow. So I joined the lab. I learned a lot of techniques also related to vascular biology. And my project was looking into how diabetes, type 2 diabetes, affects these mechanisms and affects the blood flow regulation to the brain. And mechanistically, what can we learn, and how can we target these mechanisms to protect against pathological outcomes in diabetic patients. So and this is how close I got to the brain, studying the blood vessels supplying the brain. But I was also interested in studying the brain itself, so neurons. Um, so I decided to join Steve Ferguson lab. I had a passion about G protein coupled receptors and neuroscience in general. And my interest in G protein coupled receptor came from the fact, I'm a pharmacist, so I'm licensed pharmacist. So I see a lot of drugs in the market that I dispense every day that are targeting in a way or another G protein coupled receptor. We're talking about at least 35 to 40% of the drugs that we have in the market, right? So I had like huge, as a scientist, I was really interested in studying G protein coupled receptor and at the same time studying the brain. So I joined Steve's lab because he was doing a lot of um, G protein neuropharmacology, GPCR neuropharmacology. I joined the lab and in 2015, and I've been there since. I would say it was very productive postdoctoral fellowship with Steve. Learned a lot about G protein coupled receptors. I learned a lot about neuroscience in general, and here I am today. Uh, G protein coupled receptor scientist and neuroscientist, which something that I've never thought I would be, but I guess the journey of science.
Absolutely. Thank you for that. And it's I have many questions. So you
Yeah.
went from looking at drug action in animal models, and then did a lot of physiology, and then you ended up at doing GPCRs. But before I ask my questions to around what you just told us, I have a question that goes before that. Did you ever? How did you end up in science? Were you, as a teenager, were you always interested in science, or do you have anyone in the family who's a scientist?
Yeah, that's a good question. So, I come from a family of my mom and dad are both scientists. They're both marine biologists. So I grew up in the lab. They had to take me to the lab because they had a lot of experiments to do, and I was like, I grew up going to the lab, seeing the experiments. And of course, it's a different type of experiments than what I do, but I mean, the idea is there. Their passion. I've seen their passion um, for science, and I kind of started to pick this up through the years, and the only way to do this, or not the only way, the best way to do this is to really excel in your undergrad studies so that you can um, secure like a fully funded studentship,
Mm.
master studentship in at the faculty, where then that opens opportunities for you to go anywhere or do whatever you like in terms of being a scientist. So I decided to do that during my undergrad, and I loved pharmacology, and uh, I maintained that um, love through my studies, and then when I graduated my choice for the department, because of course the faculty has different department. My choice for the department was always pharmacology, and I was lucky to join that department, and this is where I started. Yeah,
That's so interesting. I don't remember any of our guests having to both parents being scientists and then ended up
yeah,
scientists. I know of couples who are scientists, and when I ask, "Are the kids any have any interest in science?" They say, "Pff, no." But
yeah.
but that's fantastic. And then the other question I had for you is, once you finished your master's in Egypt, how did you choose to? Why did you choose to go to Canada, and what? Why not to Europe or to the US? Was that a specific choice because of the lab?
It was. So I had my eyes as that, like I was interested in the brain, and my theory as a scientist back then was kind of focused on blood vessels, because this is what I learned as a master's student, right? So
Yeah.
I wanted to study the blood vessels going to the brain. So I started to look for that. And of course, you can tell after I joined the PhD and learned more, I started to think about the brain as a, you know, as a, you know, neurons interacting with blood vessels and other cells as well. So, but back then I was thinking about the blood vessels going to the brain. What do they do? How do they work? And I started to look everywhere um, on the places where I can do um, um, you know a PhD looking into the regulation of blood vessels supplying the brain. Not a lot of labs were doing this, and there was this group, a very prominent group at the University of Calgary. It's called the Smooth Muscle Research Group. They were well known for their expertise in terms of studying blood vessels in general, and more specifically the cerebral blood vessels. So that's why I had my focus on that group, and at the same time, I had a friend who was working in the same group, and he assured me that there is um, a lot of expertise in that particular group, if you want to study this particular topic. And that's why I was I did not apply everywhere. I was looking into a certain lab in a certain area, and I was able to find it, and things worked out. I contacted Bill, and things worked out. He had grants when I contacted him, and he said, "Okay, I can have a student," and I was his last student. He did not take any students after me because he's retiring. So I was his last student. He got the grant. I was his last student. I was lucky because being last student, and pretty much for most of the time, I would say the only PhD student in the lab. I was kind of spoiled. He was spent time with me, teaching me techniques, or um, um, tips and tricks in writing and things like this. So I learned a lot from him, and of course from the group itself, because there are a lot of great scientists that are kind of focusing on the same area. And when we have meetings or journal clubs, you can see how much of a great group it is in terms of the questions and the science that they are discussing. So I was lucky with that choice for my PhD.
So it was scientific and very strategic.
Yes, I would say so.
That's great. I love that. Let me ask you a more not so scientific question. The temperature change from Alexandria to Calgary, the shock. How was that? How did you live through the shock? And then, joke aside, how easy or hard was it? How was it for you to adapt to a brand new country, a new culture? These questions I think might be very interesting to some of our young scientists listening,
Yeah,
PhD students, postdocs, and I think it's really part of the scientist's ability to adapt.
yeah,
But tell us a little bit more about your experience.
yeah. I truly believe in what you said right now, that the scientist has to adapt. It's in every step in your career, you will have to change the group we're working with, change the lab, change the university, to learn new things, to pick up more expertise for your future. So I think we are we from day one we have to learn how to adapt, and of course you can tell that in the lab. Even if you're in one lab, you can see the turnover, like people leaving and people coming back, like hiring new students, student finishing, or postdocs leaving and another postdoc coming. So it's a constant adaptation. You adapt to the team you work with. So it's a very important, I would say, important merit of the scientist to be able to adapt all the time. For me, it was like, it was really hard. I arrived Christmas Day, so I arrived Christmas Day at 4 p.m. and I leave the airport and nobody in the street, like nobody, seriously, nobody. It's Christmas Day,
It's
and I was like,
Calgary.
"Is this
And
is this normal? Is this normal? Is this how it should be?" I've never left Egypt at
yeah,
that point. It was the first time I leave Egypt, and it was like, "Oh, this will be hard." Then I was so tired, so I went to bed. Like, of course, it covered in snow, and I was excited with the snow when I landed. It's first time for me to see snow, and then next day was Boxing Day, 26. So I go out, I leave the house, and I go out, and everybody's on the street, like everybody in the mall. So it was two different experiences, like between the 2 days. But by time I got used to, I mean, if you have the you know right gear, you and you don't spend so much time outside, so it's really you can kind of adapt. And I love Canada. Canada is so welcoming. Canadian are great, so it was an easy transition for me, I would say, um, to come from Egypt and Canada. And, Canada for me is home now, right? I've been here for over 12 years, so. Um, and as I said, like, adaptation is one thing that we have to live with. Even at like I you know leaving the lab, going to another lab, as I said, staying in the same lab, you'll see a lot of people coming in and leaving. And no matter what stage of career you are, it's going to be hard. Like, if you are a trainee, you'll see your friends leaving, and
yeah,
a new trainees coming in. Even if you are the PI, like the people that you trained and you spend the time with are leaving, and then another trainee is coming. So it's hard at all. Like, so you have to learn how to adapt and keep going, because this is the life in the lab, and it's fun.
agreed, agreed. Thank you for that. Well, that's a nice image. Day
Yeah,
one, nobody. Day two, everybody.
everybody in the street. Yeah.
I love it. So, when did you first hear about GPCRs, and what drew you to go from your PhD theme in Calgary to Steve Ferguson's lab in Ottawa?
Um, of course, I've learned about GPCR during my undergrad. As I said, a lot of drugs target GPCR, so it was kind of um, highly visited topic during my undergrad. And then when I joined Bill Cole Lab for a PhD, it was around 2012 when Brian Kowalka and Bob Lefkowitz got their Nobel Prize. So I started um, to dig more into GPCR, and learn more about the mechanisms, and started to learn those new topics. To me, were new: biased agonism, biased signaling. So these things were very interesting. And I always wanted, like, I started to develop passion for these things, and I started to look for, keep my eye on people who are actually working on G protein coupled receptor. And Steve was, of course, one of the biggest names in Canada or doing G protein coupled receptors. And what I liked about Steve's lab is his approach in studying G protein coupled receptors. So he's not heavily focused, for example, on the signaling, and, for example, cell culture models, but he's doing cell culture models, he's doing animal models, he's doing, it's kind of an integrative approach to study G protein coupled receptor from in vitro, ex vivo to in vivo. So I wanted to get that kind of experience where, learn about certain mechanisms in vitro and start to check them out in vivo and see if that have any therapeutic implication. And this is what I've been doing over I would say the 6 years, where we look for mechanisms ex vivo or in vitro and validate it in vivo. And it's quite rewarding to see that there is kind of a translational meaning for what happens in cells or in culture, primary cultures, or in brain slices. So this is what I really liked about Steve's work. And that's why I kind of decided to that after I finish my PhD, I'm going for G protein coupled receptors, and of course, my passion about brain was developing then, and about neurons and how they communicate, and then so Steve was kind of a perfect lab for me to join, and this is what I've been doing, since then.
I love it. Again, very methodical, strategic, and scientific.
Thanks.
So you mentioned that you've been in Steve Lab, and I have to ask this, and I ask this from everybody. Do you have a favorite GPCR? Since you seem to be more like a physiologist and looking at, cellular models, but also ex vivo and in vivo validation of the observations. Any favorite GPCRs?
Yeah. If you just Google me, you'll find that I have one favorite G protein coupled receptors. That
I did. I have to ask.
Methylglutamate receptor 5. I love that receptor, and that. This is what I've been focusing on over the past few years, studying many of the novel mechanisms and signaling mechanisms of that receptor and how they can be how its signaling can contribute to pathology, and how can we therapeutically target that particular receptor. So I would say this is my favorite receptor.
Did you pick this project, or did you kind of build it together with Steve, or how did you?
Yes and no. I was so funny fact, when I came to Steve's lab, he had a vascular biology related project for me to start on. And this project, like another project, was like looking into how like the contribution of mGlu5 to Huntington disease pathology was kind of on the back burner there. Wasn't It was a project in the lab. They had mice and developed mice, and it was not meant to be my project. But I kind of when I joined Steve Lab and I started working on this vascular biology related project that I don't want to go into the details of this because we didn't actually continue
envisioned
with it.
Okay.
Yeah, we didn't even continue with it. But I was working on this and I saw that project that I had, I wanted to work with mGlu5. And I talked to him, and he said, "Yeah, you can just go ahead and work with that project if you want." And since then, I haven't left mGlu5, and he didn't even continue on the project I was supposed to do, and kind of started to branch out from mGlu5 to in Huntington disease to other neurodegenerative disease, and this is what I've been doing since. It was in the lab. Steve has been studying mGlu5 for a few years, and this project was promising. They had a negative allosteric modulator for mGlu5. They wanted to test whether it's beneficial in Huntington disease. Have never been tested before. The lab has shown that when you knock out mGlu5 in Huntington mouse model, you see an improvement, but they were not sure if you were able to achieve that pharmacologically. So it's kind of fits my area in different aspects. First, in LOR5, the therapeutic potential of that project as a pharmacist. So I loved it. So I just, I said I would just try. And it involved a lot of learning because I had to learn how to do all the behavioral tests, right? Like the motor function test, cognitive function test. I haven't done that before, of course. So I had to learn how to do this, along with learning how to study G protein coupled receptor signaling and the brain itself. So it involved, it was a, I would say it was a sharp learning curve, but very rewarding at the end.
Thank you. You made a couple of great points that I'd like to point out. One, I love the fact that you're, strategic. You planned ahead. You figured out what you liked, and it seems, based on what I'm hearing, the project in Steve's lab that you started, that you ended up working on, ticked all the boxes scientifically for you.
Yeah,
But then
exactly.
the great point that you also made is that you needed to learn new things
Yeah,
in order to complete that project. And I think, again, for junior scientists, for master's, PhD students, that would think that, once you've been in the field, oh my God, you know so much. Yes, well, everybody had to learn all of those things.
yeah.
And it's a steep learning curve, but it's very rewarding once you learn all those techniques.
Correct. Thank
So
you very much for saying that, because every time, like now, I think every time I'm faced with something that I should learn, I actually go ahead and learn, because I found that through the years that every single learning experience you go through, you will use it at some point. It's important. It kind of adds up to your expertise, adds up to how you think about science, how you approach science, and also adds up to your CV, which is very important, right? So now I'm in the job market, applying for positions, and people are looking into. Yeah, I have to say, when I switched from vascular biology to neuroscience, I was really worried. I thought that my career will end at this point, because I made the switch between two different areas, completely different areas, because I'm not an expert in a certain area
yeah,
from master to postdoc. So hiring will be hard, but this is not right. Like, a lot of people are really interested in that kind of expertise now, and for example, that it gives me the edge to study how there is an interaction between neurons and vascular biology, or blood vessels, right? So neurovascular coupling, for example, because I have expertise in both areas. So um, back in the days, I never thought about this, but now I think about it, because I kind of been exposed to two areas, so I want to make use of the two expertise, and develop something innovative, for example. So that I haven't done before, and a lot of people really appreciate that aspect. For example, when I apply for positions, they appreciate that aspect, that there is diversity and the expertise,
yeah.
um, not really heavily focused on one area, but kind of a diverse expertise, which is something I've never thought of, but it kind of worked out.
I love it. And again, you made another bunch of great points, which I really love to highlight is, yes, the fact that, you have a project, you needed to complete it. You learned the techniques for you to complete the project. But at the same time, learning different things from different fields is important, is
Yeah,
actually a good thing.
yeah,
And the overall experiences that you collected by working in both fields make you unique,
yeah,
make it so that not only you're a great scientist and you master those techniques, but at the same time they show something even more important: adaptability, and
yeah,
also the ability to learn.
yeah,
And oftentimes, I know from experience, I was like that. I worked on GPCRs forever,
yeah,
and there was nothing fancy about it. It was different receptors, different ligands, breadth, different signaling pathways, and I could spend my days looking at those response curves and crying how beautiful they are, and that would make me super happy.
yeah,
But what I also learned is I learned to take problems and take them apart and think not only strategically, but also build out a plan, a
Yeah,
scientific plan on how to get those answers. And that's very important on the job market, whether you're looking for academic or you're looking for industry positions, you need to be able to use that critical thinking.
correct. Correct.
Let's get back to mGluR5. Tell us a little bit more about the highlights of your work. What are you the most proud of? What are the most proudest discovery moments that you had?
Um, that's a great point. So I think the first great moment where was in 2017 when we discovered that mGluR5 can actually um, regulate autophagy, which was something that was not even described for any G protein coupled receptor. So I would say that was the first direct evidence that a G protein coupled receptor can, in fact, regulate autophagy, which has major implication, right? Autophagy is a very important mechanism that make sure that the cell gets rid of toxic cargoes and misfolded proteins and things like this. And accumulation of these things is truly toxic to neurons. So when we discovered this, it was great because if you were able to target that particular receptor, you can regulate autophagy, and that way you can get rid of some misfolded protein. And this is what we found, for example, on Huntington's disease and Alzheimer's disease. When you target the particular receptor, you can get rid of mutant huntingtin, the misfolded, the
Yeah.
mutant form of huntingtin in the brain of Huntington patient. You can get rid of amyloid beta in Alzheimer patients. So it was one of the best moment when we were able to show that link between that particular receptor and autophagy. And autophagy itself, very cool mechanism, but very hard to target because you cannot find a drug that can target that induce autophagy without major side effects or without a lot of toxic effects. But the fact that you can target autophagy or at least certain mechanisms of autophagy using a G protein coupled receptor is a breakthrough itself because the G protein coupled receptor is very easy to target, right? You can create so many ligands to specifically target certain G protein coupled receptors, and you have the choice to use agonist, antagonist, allosteric modulators. So it makes it therapeutically viable to target a G protein coupled receptor and influence autophagy, if that makes sense for some diseases like would be assumed for Huntington Alzheimer's disease. So this is moment number one. Moment number two was in 2020 when we showed that mGluR5 can actually signaling in a sex specific manner, which is again another one of the very few examples where you can actually show that the receptor itself is intrinsically different between males and females. It's not like it's regulated with like by sex hormone, which is a possibility, of course, and some people have shown this. But there is an intrinsic difference between the receptors between males and females, which means that the assumption that a drug target that, for example, target one G protein coupled receptor will have the same effect in males and females is not right anymore, because the receptor itself can be actually different. And this was a very critical point in my career, not only because it's new and it's discovery, but because it really turned my attention to a lot of things that we did not see before, we did not think about before. Because once we found that, we started to go back and look into differences between males and females in the signaling between for mGluR5 and other receptors. And believe it or not, every time you look, you try to see if there's a difference between both sexes. There is some sort of difference. Some mechanisms are not, but some mechanisms are, and we kind of missed those things. And I think it was the right time for us to go back and look at them and learn more about the differences between males and females, because this is what I now always call in the papers. I think we're going into the era of sex tailored treatments,
Yeah.
where you actually need to be sure that the treatment that you're using is actually effective in each sex, uh, without assuming that it would be effective in both sexes. So those are
Wow.
the two big moments, I would say.
Wow, that's fantastic. And I think the oftentimes when you do mouse studies, it's people will choose, only males or only females, and they're a certain age, the
Yeah,
mice. And I think, yes, we should be looking at males versus females, old versus young.
yeah,
It's hard. I mean, managing animal studies, it's very difficult.
very hard and expensive. Yes.
And exactly, hard and expensive, but I think the clues that we would get out of there, and we would be able to potentially discover targeted therapies
Yeah,
for a specific age group or a specific gender.
yeah.
That is just so amazing.
Thank
Um, and if you wanted to treat Huntington's or Alzheimer's by targeting mGluR5, what would that molecule look like? And this is purely speculation. You can wish for anything.
Yeah, if I'll build on our observations that when we block mGOR5, when we block mGOR5, we see a significant improvement in symptoms and pathology in Huntington mice and Alzheimer mice, and we see that like this increase in autophagic clearance of those misfolded proteins in the brain. So we have used a negative allosteric modulator of mGOR5, and I think this will be like, if I have to imagine ideal candidate, I think that will be the ideal candidate. The fact that we were able to see improvement is one factor. The second factor is that as an allosteric modulator, it offers a great deal of selectivity, of course, to the receptor.
Yeah.
So there is a selectivity. There is we're not able to see any side effects, and even the negative allosteric modulator have been used clinically um, for other indications, or tested clinically for other indications, and like haven't really exhibited any side effects. And it's doable. You can make a negative allosteric modulator that is orally available, because the one that we use, for example, was orally available. We're giving the mice um, the drug in chocolate pudding, so they loved it. Yeah, they loved the
Not a
chocolate
bad,
pudding. Yeah,
not a bad
nothing
delivery
else, just
system.
only chocolate pudding. So they loved it. So I mean, so the versatility of the pharmacokinetic profile of allosteric modulators, the selectivity, I think makes it like a very like ideal candidate for that drug that we have in mind.
Yeah.
So this is how I see it. I see a great potential in negative allosteric modulators of that particular receptor, in certain indications for sure.
And I have another follow-up, not kind of naive question. mGluR5 expressed in the brain, is it differentially expressed in specific neurons? In the context of Alzheimer's and Huntington's, would you want to specifically target the receptor in a certain region of the brain, or we wouldn't care, quote unquote, because we would
You
have a
you
specific
would y-
molecule?
you would ideally love to target in a region, a specific region, but that's it's very hard to achieve, almost impossible, right? When we have used the drugs in animal models,
Mhm.
and we looked at the clearance, for example, in Alzheimer's disease. So we gave the drug to Alzheimer mice, and we looked in the brain, in different regions of the brain that we know. For example, hippocampus is responsible for memory, cortex, um, through the cortex. So we see a reduction in amount of amyloid beta in the cortex and in the
Yeah.
hippocampus. So it seems like its beneficial effect is kind of happening in different brain regions. We see as well rescue, for example, in the neuronal loss in the hippocampus and the cortex. So it seems like it is working in different regions too, like in the same fashion.
Mhm.
But we still don't know whether it will have differential effect. We have evidence that, for example, some mechanisms are activated in the hippocampus and not in the cortex, and vice versa. And some mechanisms are activated in the striatum and not in the cortex, and vice versa. So it's definitely not doing the same thing in
Yeah.
all brain regions. But when we look at the overall picture, there is an improvement. So it seems whatever mechanisms are happening in each region are kind of working together to give us a very nice improvement in symptoms and pathology. So, but we still have to dig into particular mechanisms in each brain regions. That's the great point that you raised there. Yeah.
Awesome, awesome. And talking about improving or increasing autophagy. So intermittent fasting
Yeah.
is thought to increase autophagy.
Yeah.
Have you ever tried in combination in your studies to fast the mice
Oh, that's a
plus
great
minus
point.
the drug?
Yes, that's a great question. No, I have not, and
The
I
reason
have not thought about it. So,
the
but
reason
it's
I'm asking is because I was talking to John Stryker on a podcast episode,
yeah,
and so they're studying the effect of painkillers of morphine,
yeah,
and they saw with the same they needed less painkillers in mice that were uh, intermittently fasted.
okay.
So that might increase the effect. I don't know what the exact mechanism, but I was wondering if that could be a good experiment to
A,
think about.
a possibility, of course, and good experiment actually to think about.
I
I've never thought about it, to be honest, but it's a great point. I mean, if we were able to induce autophagy or induce the protective mechanisms by any means, I'm sure we'll have the drug action in
Yeah.
a way or another. But it's a great point, yeah.
Hopefully you'll get to present the data at the next GPCR
Hope
retreat.
so, yeah. Hope so.
All right, thank you, Khaled. We've talked a lot about advice for junior scientists about adaptability and really being strategic, but at the same time being open to learning new things. Any advice to impart in
Um,
addition to that?
I yeah, I think that all the questions that you asked um, raised great points, and you raised great points through the interview. Um, but I think it's one important advice for me to young scientists. I'm not old, but I am, kind of
You
older,
you,
right?
you have a
Uh,
PhD, you've done a postdoc, so
yeah.
in
So
that realm, yes, you're
yeah,
experienced. I'm not gonna say old. I'm just gonna
yeah.
say experienced.
So from my limited experience, I would say follow your passion. It's very important to follow your passion. Um, don't do things because there's nothing else to do. Um, because the more when you have passion for science, you'll be able to be very successful. You'll be able to um, um, prove um, prove that you can do science um, in the best way possible, and at the same time, that will open a lot of possibilities for you in the future in terms of jobs. It doesn't have to be academia, but other jobs as well. So,
Yeah.
for if science is if you think it's not meant to be for you, there's nothing wrong with this, right? You can have, you know. Um, uh, you can do something else, but the
Yeah.
most important thing is to have passion for science, because um, the amount of frustration in that job is huge, and if you don't have this passion, you will not be able to continue. Just every day, look, of course, like every day you go to the lab, there is some sort of experiment not working, or are and optimizations that need to be done, and you spend months and on months optimizing a certain assay, and then it doesn't work out, and things like this. So, or you send a paper and it comes with a rejection, or it comes with, you know, um,
3 years of experiments to redo.
unreasonable experiments to do, or you submit a grant and it gets rejected. So all these things, we are in a constant phase of rejection in experiment, in papers and grants. So if you don't have that passion, you'll not be able to wake up in the morning and go and do that again and again and again until you get it. Which is important. If you don't have that passion, then you'll be depressed. So you'll not be able to do it. Right?
Yeah, I think that's a great wake-up call to do something else. But I think we all live for that dopamine rush, for the time when the experiment works, or for the time when you get the email saying congratulations, your grant was funded, or your paper was accepted with minor modifications, or and then we go from one dopamine rush to another, but the distance between the dopamine rushes is quite long.
Yeah, I agree. So and you'll get it. You will get the grant. You'll get the paper published. It will happen. It just needs you need that passion, and need the patience to get these things done, right? So
agreed.
Yeah.
If you don't mind me asking, what's next for you? You mentioned job search. You don't have to answer. You don't have to answer.
It's all good. There's nothing wrong. I'm job search, so in the phase of interviews and things like this. So hopefully something works out.
Academia,
yes.
Awesome.
Academia.
Awesome.
So hopefully something works out, and of course, you'll be the first to share with. I will share with you the news if it happens.
Thank you. Thank you. Excited for that. With this, I want to thank you for your time. I really am happy that you were able to talk to me today, and I wish you happy holidays, happy New Year. For those of you listening, when this episode is going to come out, they'll know that we recorded this in December.
Yeah,
but that's okay. Thank you so much, Khaled.
Thank you very much. It was great. We appreciate this fantastic interview and talking to you, and hopefully we'll get to meet very soon in one or another meetings, of course. So hopefully it will happen next year. I hope so.
Yeah, I have my fingers crossed for the Niagara on the Lake GPCR retreat in May. Hopefully it'll happen.
I hope so. They said it will happen, but I'm not sure what will happen now.
I don't know. Maybe we're gonna arrive there with the suits like in the P4.
Yeah.
With the oxygen tanks behind us and
possibility. If this is what it takes, let's do it.
Yeah, absolutely. All right. Thank you so much.
Thank you very much.
Thank you for joining us and listening to this podcast episode. We'd like to thank our guests, as well as our team members, Attila Forrest and Inés Pinheiro. Please subscribe to the Dr. GPCR newsletter, find us on YouTube, and if you like our podcast, leave us a 5-star review on Apple Podcasts. You can also leave us a testimonial at drgpcr.com/testimonials. Another great way to support us is to share your favorite Dr. GPCR program with your network and colleagues. Stay tuned for the launch of our brand new project in the upcoming weeks, and email us with any questions or suggestions at hello@drgpcr.com. Until next time, stay safe.